Okay, so welcome back to this next video in which we are discussing voltage-gated ion channels. Okay, right, so we're in the process of discussing voltage-gated potassium channels in more detail. Okay, and we've seen how voltage-gated potassium channels are constructed out of 4 alpha subunits of voltage-gated potassium channels. Okay, and we've looked in detail at the structure of an alpha subunit of a voltage-gated potassium channel. We've looked at the basic domains that they all have. Okay, so T1 tetramerization domain and all of these membrane spanning domains. Okay, and we've then seen how those different membrane spanning domains are arranged in the voltage-gated potassium channel tetramer as so. Okay, right, so what I want to do now in this video then is talk about the diversity of voltage-gated potassium channels, okay, because the difficulty here is that it would be beautifully simple if there was just one alpha subunit of a voltage-gated potassium channel, and we could call it the alpha subunit of voltage-gated potassium channels, okay? And then all you had to do to make a voltage-gated potassium channel tetramer was make 4 of these alpha subunits, stick them together, and you'd have your voltage-gated potassium channel, and all voltage-gated potassium channels in the human body would be identical. That would be the beautifully simple scenario. Okay, unfortunately it is not the case. Instead, there are 40 different genes which all code for voltage-gated potassium channel alpha subunits. Okay, so 40 genes coding for 40 different alpha subunits of voltage-gated potassium channels, and that isn't even counting the different splice variants, okay, which some of these do have, but we're not even going to go there with the different splice variants. Okay, it's just enough of a hassle to talk about the 40 different genes. Okay, so there are 40 different genes coding for alpha subunits of voltage-gated potassium channels. Okay, and all of these 40 different genes will give rise to alpha subunits of voltage-gated potassium channels which have different fundamental sequences of amino acids. And so the alpha subunits created by these different genes are going to have different primary sequences of amino acids. However, they will all have those same basic domains that we have talked about. So they'll all have a T1 tetramerization domain, and they'll all have those basic membrane spanning domains that we've talked about, even though their primary sequences of amino acids all vary, basically. Okay, so all of the structural stuff we've just been talking about, that applies for all voltage-gated potassium channels, even though they all have, well, they're all going to be constructed out of different alpha subunits, which fundamentally have different primary sequences of amino acids. Okay, right, so now what I want to tell you about is the classification of the 40 different genes for voltage-gated potassium channel alpha subunits into 12 different families, because there are 40 genes, but they're grouped to help us understand into 12 families, basically. And then I want to tell you about how you can assemble tetramers, basically, because of course we're talking about the individual alpha subunits. We're talking about the fact that there are 40 different genes for alpha subunits of voltage-gated potassium channels. Okay, so there are 40 different subunits of voltage-gated potassium channels. How many different tetramers does that mean that you can actually make? Well, the answer is phenomenally huge number of different tetramers you can build. Okay, now I'm going to tell you about the ones that have actually been found in humans. Okay, so the main sort of tetramers that you can actually build, basically. Okay, but first, let's talk about the classification then of these 40 different genes for alpha subunits of voltage-gated potassium channels into 12 different families. Okay, right, so let's start off with the nomenclature for the 12 different families. Okay, so the 12 different families then are called KV1, KV2, KV3, all the way down to KV12. So what does this mean? So here's the name of the first family of alpha subunits of voltage-gated potassium channels, basically. Okay, now what does this stand for? Well, the K obviously stands for potassium, and then the the subscript V stands for voltage-gated. Okay, so that's the nomenclature here. Okay, so there is this KV1 family, which contains some genes for alpha subunits of voltage-gated potassium channels. Okay, and this KV1 family is actually the biggest family of voltage-gated potassium channel alpha subunit genes. Okay, this contains 8 different members, okay, which are called KV1.1, KV1.2, KV1.3, all the way up to KV1.8. Okay, so 40 genes in total. Here are 8 of them already, just in this first family of genes for alpha subunits of voltage-gated potassium channels. Okay, and those first 8 are called KV1.1, KV1.2, KV1.3, KV1.4, KV1.5, all the way up to KV1.8. Okay, so that's the KV1 family of genes for alpha subunits of voltage-gated potassium channels. Next up, let's have the KV2 family of genes for alpha subunits of voltage-gated potassium channels. This is a much smaller family than the KV1 family. This only consists of 2 members. Okay, specifically there is the KV2.1 gene, and then after that there is the KV2.2 gene. So the naming of these is very, very logical. Okay, so it's very, very nice. So there are 2 members in this KV2 family, okay, and that takes us up to 10 of the different genes already done, so only another 30 to go. Okay, so let's continue on with the families. Okay, next up, of course, we'll go on to KV3 family. Okay, and the KV3 family contains 4 different members. Okay, so there is KV3.1, and that goes on all the way up to KV3.4. Okay, so we're now up to 14 different genes that we've covered now. Okay, another 26 to go. Okay, next up we'll have the KV4 family. Okay, and the KV4 family contains 3 different members. Okay, and again, the nomenclature is always going to be this very sensible nomenclature. Okay, so it's KV4.1 all the way up to KV4.3. Now, I probably should say that a lot of these alpha subunits for voltage-gated potassium channels, they have old names as well. Okay, and we're not going to go obviously through the old names for all the different alpha subunits of voltage-gated potassium channels. That would be a nightmare, but be aware that these are the sensible names that people have now agreed that these things should be called, but many people will still use the old name. Okay, so when you actually meet these in some physiological context, you will probably learn their old names as well. Okay, but try and also learn their new names to fit it in with what I'm telling you about voltage-gated potassium channels. Okay, so there are 3 different subunits then in this KV4 family, so that takes us up to 17 different genes that we've now covered. Okay, in the KV5 family there is only 1 member. Okay, so that's just called KV5.1. Okay, so that's a nice simple family. Okay, next up onto the KV6 family of voltage-gated potassium channel alpha subunit genes. Okay, and the KV6 family contains 4 members. Okay, called KV6.1 all the way up to KV6.4. Okay, and we'll underline the KV6 family, I think, in yellow. Okay, then I might go over to here now. Okay, so next up we've got the KV7 family. Okay, and that will consist of 5 members, so this is the second biggest family after the KV1 family. Okay, and this contains KV7.1 all the way up to KV7.5. Okay, and we'll underline that family in green here. Next up, the KV8 family, and this is another tiny one now. Okay, this only consists of 2 members, KV8.1 and KV8.2, and we've lost count of how many we've gone through now. Okay, and it should have just been a comma there rather than a dash. Okay, and I'll underline the KV8 family in pink here. Okay, so let's just recount up where we got to. So 8 here, 2 here, 10, 14, 17, 18, add on 4 there, 22, 5 takes up to 27, this takes us up to 29, so we've got another 11 to go basically. Okay, so next up we'll go on to the KV9 family. Okay, so the KV9 family has 3 different members in it. Okay, KV 9.1 all the way through to KV 9.3. That takes us up now to a total of 32 different genes for alpha subunits of voltage-gated potassium channels, and we'll have to now start recycling the colours because I've only got 8 different colours. So now we're back onto orange. Okay, next up we'll have the KV10 family. Okay, and the KV10 family is again just a small family, KV 10.1 and KV 10.2. That takes us up to 34 total alpha subunits of voltage-gated potassium channels. Okay, next up we'll have the KV 11 family, okay, which consists of 3 members, KV 11.1 all the way up to KV 11.3. Okay, that takes up to a total of 37, and now we've only got another 3 more to go, and those are the 3 in the KV12 family. Okay, so the final family is called KV12, okay, and this consists of 3 different members, KV12.1 through to KV12.3. Okay, and we'll underline that one in blue here. Okay, right, so those are the 12 different families then of alpha subunits of voltage-gated potassium channels, or rather the genes for alpha subunits of voltage-gated potassium channels. Many of them have known different splice variants, which adds to the humongous great complexity of voltage-gated potassium channels that we are capable of building. Okay, right, so these are the genes for the individual alpha subunits. Okay, what I now want to talk about is what sort of tetramers can you actually build? What sort of voltage-gated potassium channel tetramers are you actually allowed to build? And because you will appreciate the horrendous capability here that we have for a huge complexity, okay? Because, you know, you could combine potentially any of these together, okay? You could mix and match completely. You could pick 4 different ones, combine them together, and make a voltage-gated potassium channel. However, the ones that are actually found in the human body are a lot simpler than that, basically, okay? So I'm now going to tell you about the tetramers that we actually know exist. Okay, so firstly let me tell you about the simplest type of tetramer. So the simplest type of tetramer that you could imagine is a homotetramer. Okay, and a homotetramer, as the name suggests, is just a tetramer where all of the 4 subunits that make the homotetramer are exactly the same. So homo means the same. Okay, so the 4 subunits that you're going to use to build your tetramer are going to be from the same gene, basically. They're the same type. Okay, now you can do this. You can build homotetramers for every single one of the alpha subunits of voltage-gated potassium channels, apart from some which lie in certain families. Okay, so you can't build them for absolutely everyone. However, most of these alpha subunits can form homotetramers. So which families are the ones that can't actually build homotetramers? Okay, so the families which are not capable of actually forming homotetramers are the KV5 family, so that's an easy family, that only contains 1 member. So KV 5.1 cannot form a homotetramer. You can't put 4 KV 5.1s together and actually build a functional voltage-gated potassium channel. Okay, the KV6 family also, so none of these 4 genes here in the KV6 family can form homotetrameric voltage-gated potassium channels. Okay, and what do I actually mean by that? How do people know that you can't build homotetramers out of these types of alpha subunits of voltage-gated potassium channels? Well, quite simply what you do is you put the gene into a type of cell that doesn't actually have any genes for voltage-gated potassium channels in it. And then you see, does the cell actually actually make voltage-gated potassium channels if you just put that gene in. And if it makes voltage-gated potassium channels, then it must be the case that it's making these alpha subunits for that gene and then assembling them into homotetramers, and they must be homotetramers if they're being produced, because that's the only gene that the cell then has for the alpha subunit of a voltage-gated potassium channel. So that's quite simply how you find this out. Okay, and we've done this for the one gene in the KV5 family, and it doesn't work. You don't get functional voltage-gated potassium channels if you just put the KV5.1 gene in, and neither does it work for any of the genes in the KV6 family. So all four of those don't form homotetramers. And the list goes on as well. KV8 family and KV9 family are just as bad. They don't form homotetramers. None of the genes in those families form homotetramers if they're expressed on their own, basically. Okay, so these families, their genes in these families are not capable of actually producing proteins which can then form homotetramers. Okay, so the genes, or rather the alpha subunits within these four families here, are what are known as modifier subunits. Okay, they have to be put in heterotetramers instead. Okay, so another key word here. So we've had homotetramers, which are tetramers where the four subunits that make up the tetramer are identical. Okay, now we're going to have the concept of a heterotetramer. So hetero means different, tetramer means four-membered structure. So a heterotetrameric voltage-gated potassium channel is one where the four different alpha subunits that make that voltage-gated potassium channel are different types of alpha subunits of voltage-gated potassium channels, basically. Okay, now the genes then in these four different families here, the genes in KV5, KV6, KV8 and KV9 then, they have to form heterotetramers with alpha subunits from another family, and specifically this other family is KV2. Okay, so let me explain this in detail. So basically, I've said that if you take any of these genes from any of these four families, okay, and you express it and you put the gene in a cell on its own, and the cell doesn't express any other alpha subunits of voltage-gated potassium channels, then you don't get functional voltage-gated potassium channels being produced. If, on the other hand, what you do is you take one gene from one of these four families here, and you put it into a cell, okay, which doesn't have any other alpha subunits for voltage-gated potassium channels, and you also put in a gene of one of these KV2 subunits here, okay, so either KV2.1 or KV2.2, then you do get voltage-gated potassium channels being produced. Okay, so what happens basically is the alpha subunits produced by the genes in these four families can form heterotetramers, okay, tetramers where there are other subunits in there as well, and these other subunits are KV2 subunits. Okay, so you can build heterotetramers of any one of the genes in these four families with any one of the KV2 subunits, basically. Okay, so that's why these are called modifier subunits, because they are compulsory additional subunits that can mix with the KV2s, basically. Okay, so all of the other genes, beside the genes that are in these four families then, they can form homotetramers. So the alpha subunits they produce can form homotetramers. Okay, so there are a huge number of different homotetramers there. So for instance, all of the KV1 alpha subunits, they can all form homotetramers. All of the KV2s can form homotetramers, and onwards you can take this on. Okay, right, so there's a whole bunch of homotetramers that you can create. Now I've told you also about these special heterotetramers, which are heterotetramers of alpha subunits from the genes in these four families that can't form homotetramers with alpha subunits from the KV2 family, basically. Okay, I'd now like to tell you about some other heterotetramers that are known to form. Okay, so it's also known that you can form heterotetramers within families, okay, specifically within the KV1, KV7, and KV10 families. Okay, so what do I mean by this? Okay, so basically we've seen that these families here, Kv1 family, Kv7 family, and Kv10 family, they are not in these modifier subunit families. Okay, so all of the genes in these three families are capable of producing alpha subunits which can form homotetramers, basically. Okay, however, you can also form heterotetramers from the alpha subunits in these three families. However, the heterotetramers are only mixed and matched between the family. Okay, so what do I mean by that? So basically, I'm saying that you could make a heterotetramer, for instance, involving Kv1.1 alpha subunits and Kv1.2 alpha subunits. Okay, so you could have a heterotetramer which contains some Kv1.1 and Kv1.2 subunits. Okay, because they are both in the same family. You could do it with Kv1.1 and Kv1.8 because they're in the same family. Okay, so the alpha subunits that are in the same family can form heterotetramers in the case of the Kv1 family, the Kv7 family, and the Kv10 family. So in those three families, you can form heterotetramers which are within the family, so intra-family heterotetramers, basically, where you mix and match the different subunits within that same family. However, we think that these heterotetramers are only heterotetramers involving two different types of alpha subunits. Okay, so we don't think you pick, for instance, 4 different alpha subunits in the Kv1 family and mix those together. It seems that it's just 2 different types of alpha subunits that are put into one tetramer. Okay, so maybe you'll put 2 Kv1.1 alpha subunits in and 2 Kv1.2 alpha subunits in, rather than 1 Kv1.1, 1 Kv1.2, 1 Kv1.3, and 1 Kv1.4. So these subunits, these heterotetramers that are within families seem to be just heterotetramers that involve 2 different types of alpha subunits. Okay, right. So that then is the huge number of different alpha subunits of voltage-gated potassium channels now covered, and a little bit of the insight into how they can actually assemble then into tetramers. So an awful lot of them then can assemble into homotetramers. Okay, and the homotetramers are incredibly important. Some of them can't assemble into homotetramers and have to assemble into heterotetramers with these subunits from the Kv2 family. Okay, and then we've also talked about these intra-family heterotetramers which can form. Okay, right, so now we're going on to a really important topic then. Okay, so I now want to discuss with you the opening mechanism then for the voltage-gated potassium channel. How does this thing open in response to a depolarization in the electrical potential difference across the cell membrane? Okay, opening in response to voltage changes, okay, or in response to depolarization. So the first thing that I need to tell you if we're going to discuss opening in response to depolarization is what is normally keeping the channel closed. Why can potassium not normally actually cross through the pore of the voltage-gated potassium channel? Okay, so we need to discuss how is the gate closed before we can discuss how to open the gate, basically. Okay, what even is the gate? Okay, so going back to this picture that I have of the voltage-gated potassium channel tetramer. Okay, so we've discussed that if you're a potassium arm moving through this voltage-gated potassium channel, if you're going from the extracellular aspect into the intracellular portion, okay, you will start off in this very narrow portion of the pore, which is the portion of the pore that is lined by these ascending portions of the P-loop here. Okay, that special portion is going to be called the selectivity filter. Okay, so the portion of the pore that is lined by the ascending portion of the P-loops is called the selectivity filter. Okay, and as the name suggests, that's the portion that's going to actually select for potassium ions over other types of ions, and we will come on to the selectivity filter later on. The selectivity filter is not believed to be where the gate is. Okay, so the selectivity filter is always open, basically. Okay, so where is the gate state then? Well, let's continue on through this pore. Okay, so if we get through the selectivity filter, if we get through this narrowest portion of the pore, which is the portion of the pore lined by these ascending portions of the p-loop, then we'll get past that, and now what does the pore become lined by? Well, I said it becomes lined by the S6 helices, and the pore is much wider down here. At least it's much wider when it's open. However, when the voltage-gated potassium channel is not in the open state, when it's closed prior to depolarization, it's not wider down there. Instead, the S6 alpha helices actually all bend inwards. Okay, so although I'm showing them as in this picture as though they're going straight down, they don't go straight down in the actual closed state channel. Instead, they bend inwards towards the intracellular aspect and close that off. Okay, so that's what keeps the pore closed usually. The internal aspect of the pore is closed by the S6 helices all sort of moving towards the centre and closing the pore so that potassium ions can't move through basically. Okay, and therefore if we want to open the voltage-gated potassium channel, we need to pull those silly S6 helices backwards basically and open up the internal aspect of the pore. Okay, right. So what I now want to draw then is a simple picture where we're taking a cross-section of the pore domain of the voltage-gated potassium channel. Okay, so what we're going to do is chuck aside the voltage sensing domains. I'm not going to draw that. All I'm going to now do is basically take a cross-section through this pore domain here. So I'm going to basically cut through it, and we're going to look through on this new opened up surface basically, and that will allow us to see the selectivity filter and then where these S6 helices are moving inwards basically. Okay, right, so let me draw this picture. So here, this is the cell membrane here. Okay, and now let me put the picture of it at the moment, the closed state channel here. Okay, I'll draw it like so. So here is the selectivity filter portion of the pore at the top here. So this is the selectivity filter here, this narrow portion of the pore, and this internal portion after the selectivity filter, so after the ascending portion of the p-loop, should be much wider if it was in the open state, but it's not at the moment because the S6 helices are moving in towards the center. Okay, so let me just bring back up this picture of an individual subunit of a voltage-gated potassium channel. Okay, so here comes back this picture. Here's the p-loop, here's S6, the C-terminal there, here is the T1 tetramerization domain. Okay, and let me now just colour in some domains here. So let's have the ascending portion of the p-loop coloured in yellow here, and here this is where the ascending portion of the p-loop is lining the pore, and it's responsible for this very narrow portion of the pore, which is going to select for potassium ions. And I've said that that narrow portion of the pore here is called the selectivity filter. Okay, however, even though that's narrow, that is wide enough to conduct a potassium ion at the moment, even in this closed state. So this is the closed state of the channel. Okay, so potassium can move through that, that's open all the time. Okay, however, this portion down here is not open at the moment. Okay, and this is because the S6 membrane spanning alpha helices are moving inwards. So I've now coloured the S6 membrane spanning alpha helices in blue here, and now I've shown that these are the portions that are moving inwards. Okay, and of course you'll have 4 S6 membrane spanning alpha helices lining the internal aspect of the pore, and they've all moved inwards to block the internal aspect of the pore, and that is why the channel is at present in the closed state. Okay, so these S6 helices being sort of moved inwards at the moment and blocking the intracellular aspect of the pore is what's known as the V-gate. Okay, so this is a gate of the channel because it's capable of opening and closing. If we were to pull the S6 helices outwards, of course that would now be the open gate, and if they're, as they are currently, i.e. moved inwards, then they're in the closed state, so that's the closed gate. Okay, and the reason it's called the V gate is because it's going to be opened and closed by voltage changes, basically. Okay, right. So now what I want to show you is the open state of the channel, and then we'll discuss how you actually get to the open state of the channel. So in the open state of the channel, this will have changed basically. You will have moved the S6 helices outwards, and instead what the channel will actually look like from the side is something like this basically. Okay, or rather in this cross section. Okay, here is the selectivity filter lined by the ascending portion of the p-loop, and here now this is the S6 helix, and we could continue it all up there actually, which I might do, because remember it is sitting behind the ascending portion of the p-loop. Okay, so now we've moved the S6 helices outwards so that they're no longer bent inwards and are no longer blocking the pore. So now we've got this nice wide intracellular aspect of the pore here. We've got the selectivity filter, which is of course still narrow, but that's still wide enough to conduct potassium ions, and now the channel is in the open state. Okay, so basically to move the channel from the closed state to the open state like this, all we need to do is pull those 4 S6 helices outwards. Okay, so why is this going to happen in response to a depolarization of the electrical potential difference across the cell membrane? What then is the gating mechanism? How do we open the V gate in response to a depolarization of the membrane voltage? Okay, well this is now where we have to bring in the voltage sensing domains. After all, they are the voltage sensing domains. Why would they be called that if they weren't actually going to sense voltage? Okay, so it's not the pore forming domains, which we've just been studying, that actually sense the change in the electrical potential difference across the cell membrane. It's these 4 domains here that are actually going to do the sensing of the electrical potential difference across the cell membrane changing. Okay, specifically it's the S4 helix which is really important. So we're now going to study the S4 helices, which are these orange ones here. Okay, so basically the S4 helices have lots of positively charged residues in them, 4 or 5 positively charged residues. Okay, and these 4 or 5 positively charged residues that they have are going to be arginine and lysine residues. Okay, so let me explain why arginine and lysine residues end up positively charged. So I'll start off by drawing the structure then of arginine. Okay, right, so I will draw this as an amino acid residue, which means I'm showing it as though it's bound within a protein rather than showing the free amino acid basically. Okay, so here is the amino group of the arginine residue, and it will be bound to the carboxylic acid group of the next amino acid along. Okay, here then is the alpha carbon with a hydrogen coming off it. Here is the carboxylic acid group here. Okay, now that's the core amino acid structure. The bit that we're actually interested in is the R group of the arginine. Okay, and the R group of the arginine, doesn't consist like that at all. Okay, get rid of the 3 and replace it with a 2. It firstly has 3 methylene groups. I was putting the 3 in the wrong place. Okay, so it has 3 methylene groups. Methylene groups are just carbons with 2 hydrogens coming off. Again, because I don't have space to draw those methylene groups actually out, I've just shown them as CH2 and then put brackets around it and then put 3 there. That means have 3 methylene groups. Okay, and then after the 3 methylene groups, what you'll then have is a nitrogen atom which will have a hydrogen coming off it, and then also a carbon atom coming off it. Okay, and this carbon atom then has an amino group coming off it up here, and then also a double bond to a nitrogen, which then has a hydrogen here. Okay, so this is the R group of arginine, and as of yet there is no positive charge there, so why does this residue end up positively charged? Well, the answer is that under normal physiological pH, okay, so under normal physiological free proton concentration, you end up getting protons adding on to this molecule, specifically this nitrogen here, which is double bound to the carbon here, has a lone pair of electrons. Okay, this lone pair of electrons here, okay, and that lone pair of electrons is a beautiful center of negative charge. Okay, protons are positively charged particles. They absolutely love to associate with centers of negative charge. So what ends up happening is that you get protons attaching to the lone pair on the nitrogen that is double bound to the carbon of arginine residues. Okay, so under physiological pH, you end up with most of the arginine residues actually having protons attached to them, basically. And this then gives the entire arginine residue a positive charge, basically. Okay, so it's a very similar story for lysine. Lysine also has a nitrogen that can accept a proton from free solution, and which will end up accepting a proton from free solution under physiological pH. Okay, so let's just talk about this then. So the R group then of lysine, well, firstly actually I'll just draw you the core amino acid structure. So here is the core amino acid structure once again. Okay, and now the R group of lysine consists of this time 4 methylene groups, and again I don't want to have to draw out 4 methylene groups, so I'll just show it like so. Here's 1 methylene group, repeat it 4 times. Okay, and then the R group, the final group on the end of the 4 methylenes is then an amino group like so. Okay, and this nitrogen here in this amino group again has a lone pair of electrons, and onto this lone pair of electrons you can get a proton attaching. Okay, so here is a proton attaching onto that lone pair of electrons, and under normal physiological pH, lysines often do end up protonated like this. So the S4 membrane-spanning alpha helix of these alpha subunits of voltage-gated potassium channels has lots of these arginines in and a few lysines in. Okay, so lots of positively charged residues, basically. Okay, and generally it has 4 or 5 positively charged residues within it. Okay, so the key thing to take from that is that the S4 helix is positively charged. Okay, so these orange helices here, they are positively charged, so I'll put a little positive here. Now, this then is how you're going to get some sort of conformational change in response to depolarization of the electrical potential difference across the cell membrane. So let's think this through. Okay, we are going to depolarize the electrical potential difference across the cell membrane. Okay, we are going to take it from, let's say, negative 65 millivolts up to negative 40 millivolts. Okay, that means that the amount by which the electrical potential intracellularly is lower than the electrical potential extracellularly has gone down. So initially, remember, the electrical potential extracellularly we can show up here, okay, and the electrical potential intracellularly we can show down here. Initially there was a drop of negative 65 millivolts. We have now reduced the drop so that it's only negative 40 millivolts. Now let's think about what the S4 helices will be doing when we're at this resting electrical potential difference of negative 65 millivolts. Okay, so when we're at the normal resting electrical potential difference across the cell membrane, what are the S4 helices going to be feeling? Well, remember, they are sitting in the membrane, okay, and they are positively charged. What do positively charged things want to do? They want to go where the electrical potential is lower, okay, which is currently the intracellular compartment by 65 millivolts. So these S4 helices are all going to be feeling a yank down to the intracellular compartment, basically. So all of these S4 helices are going to be pulled down at normal resting membrane potential difference towards the intracellular compartment. They're all being pulled down, basically, here. If we show it in this picture, all of the S4 helices are being pulled downwards, and they will pull the entire voltage sensing domains a little bit downwards. Okay, now, when we then raise the electrical potential difference across the cell membrane up to threshold potential, up to negative 40 millivolts, the strength of that pull down that the S4 helices are feeling is going to be hugely reduced. Okay, of course it's not gone. At negative 40 millivolts, the intracellular compartment still has a lower electrical potential than the extracellular compartment, so the S4 helices will still be feeling an attraction to the intracellular compartment, but the force of that attraction is reduced. Okay, so it's almost as though you've released the spring a little bit. The amount that you are pulling these s4 helices downwards is now less strong. Okay, so what's going to happen? The s4 helices are going to end up bobbing upwards a bit. You're going to get a little bit of a recoil, okay, like a spring basically being let go. Okay, we're now pulling on the s4 helices downwards with less strength, so they're going to move up a little bit. Okay, so all of the voltage sensing domains move up a little bit when you depolarize the electrical potential difference across the cell membrane. Now, what that ends up doing is pulling on the S4, S5 linker here. Okay, so all of the S4, S5 linkers get pulled on, and it's thought that what ends up happening is when the voltage sensing domains all move upwards, the S4, S5 helices, or linkers, whatever you want to call them, they are alpha helices. Okay, so S4, S5 helix, S4, S5 linker, they're the same thing. It's thought that these things sort of get pulled outwards, like so. Okay, so as these are pulled upwards, it's thought that these sort of get pulled outwards, basically. And when these pull outwards, they pull outwards on the S5 helix, which then seems to allow the S6 helix to also move outwards. So it's thought then that what you end up getting is all of the S6 helices moving outwards in response to this. And that's exactly what we now want, okay, to turn our pore from this closed state where the S6 helices are all moving inwards on the intracellular side to the open state where they're all more peripheral, basically. Okay, so it's thought that through this movement of the S4 helices outwards, you end up getting a movement of the S6 helices outwards, and that conformational change is communicated from the voltage sensing domains to the pore forming domains by the S4, S5 linker. Okay, right, so that now is the gating mechanism, the way that the V-gate is opened in response to depolarization of the electrical potential difference across the cell membrane. In the next video, what we will turn our attention onto is the mechanisms of permeation of potassium ions through a voltage-gated potassium channel. And we'll then talk about how you select for potassium ions.